Hello, everyone, and welcome to today's webinar, Accelerating Strain Development Process for CDMO and CRO Companies. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Eric Abbate, Director, Applications and Development at Inscripta. Eric, you may now begin your presentation.
So today I'm going to tell you about a high-throughput genome engineering platform that we've developed at Inscripta and how it can be used to accelerate strain development for CDMO and CRO companies. So first of all, what are CDMOs and what are CROs? So a CDMO is a contract development and manufacturing organization. And this provides really a comprehensive service all the way from research and development to commercial manufacturing. So oversees a large part of the product development process. A CRO is a contract research organization, and this provides R&D services or outsourced R&D services. So both CDMO and CRO companies are very important to the bioeconomy, particularly in recent years where companies are moving towards more of a leaner business model. And because of this, it requires much greater reliance on external partners to do certain aspects of the product development cycle. And this importance has really come out of the explosion in the biotech space. And this has led to a dramatic increase in the number of products in development. And this is not only true for larger companies that are also testing out or trying to develop many more products than they have in the past to have many more opportunities for success, as well as dramatic increase in small biotech companies that have been popping up over the years. Companies are interested in managing their R&D costs, again, towards this leaner business model, and then also to address capacity constraints. So even large companies who have their own internal manufacturing capability, due to the number of products they're trying to develop at once, they don't have the necessary capacity to do scale-up testing for all those. And then also to have access to innovation. So in biotech, there's continuously a dramatic increase in innovation and technologies that are coming out. And companies want to have access to that innovation, but they don't necessarily want to bring that innovation in-house. So they will outsource some research, R&D that needs to be done to outside companies who have expertise in this new areas of innovation. Also, for small companies, in-house development of scale-up capacity is very resource intensive, and it can also cause a shift in focus away from some of the core competencies of these small startup companies. But these large companies and small companies need to have access to productive and efficient strains that can be scaled up. So there's a lot of challenges in developing these production strains that can be used for manufacturing various different products. And when I discuss this right now, I'm going to focus mainly on, say, protein production, but this can also be applied to nucleic acid production or even small molecule production. And there's really two areas that are sort of challenging for production strain development. And they kind of break down to either yield or manufacturability. So on the yield side, you can have issues such as low expression or no expression of your target protein or small molecule. Low solubility of the protein. This is often due to incorrect folding or misfolding of the protein. You could have degradation by host proteases that are expressed by the production strain. The protein or small molecule could have some toxicity towards the production host. You can have secretion issues if the protein is being secreted from the organism, so it can get tied up in the secretion pathway. You can have inactivity of the enzyme. And this could potentially be due to post-translational modifications that might be important for the overall activity. On the other side, you have things that are related more towards manufacturability. So yield is important, but it's not everything for manufacturing. So you have to have a production strain that is tolerant to the product, whether it be some sort of enzyme or a small molecule. If you're working with bacteria, you care about endotoxin levels in that final product. So this is particularly important in the biopharma space or in the food area. So you might want to develop strains that have lower endotoxin levels. Again, for bacteria, phage resistance, so phage contamination can be a huge problem that can basically destroy a production run. So developing strains that are more resistant to phage would be beneficial. Ultimate product stability, so after manufacturing and downstream processing, you care about having a product that is stable, and changes to the host strain can have an impact on that product stability. And then also you need to have a strain that has good fermentation properties. So it basically grows well in fermentation tanks. So these are kind of some of the main challenges from a yield and a manufacturability point of view. And several of these challenges can really be overcome by scalable host strain engineering. So to optimize the development of production strains, we really need high throughput tools to make diverse edits genome-wide. And there's a lot of tools that have been developed over the last several decades that allow for strain engineering to take place. And if I plot some of those tools that are developed and put them on this axis or this matrix over here, where on the y-axis you have a variety of edit types from low to high, and on the x-axis the number of genomic locations from low to high. Simple things like gene knockouts or gene knock-ins is a simple edit type, and also you're targeting maybe one location. Something like classical mutagenesis, where you treat a production strain with a chemical mutagen, you can introduce tens to hundreds of different edits or changes within the genome, but it's a very low variety of edit types that you can introduce. If you're doing something like classical protein engineering, in this case, you're targeting maybe a single enzyme, and but you can have a wide variety of edit types. So few number of genomic locations, but a high edit type. And what you really want to do in order to scalable genome engineering is to get up in this quadrant over here, targeting a large number of genomic locations with a high variety of edit types. And really CRISPR genome editing has allowed you to get up into this quadrant, but in order to have its full potential, you need to have a tool to build very large libraries in a high throughput manner. And towards that end, I would like to introduce you to Inscripta's Onyx Platform, which allows for programmable and trackable variation to be introduced into E. coli and yeast, so the two microbes that we are currently supporting on the platform. And the Onyx Platform really supports all areas of the design, generate, test, learn cycle. And this is really the basis of the scientific method. So it all starts in design, where we have an easy-to-use web portal for design and ordering of editing libraries. And this is the software package is called Designer, and it allows a customer to go in and design whatever changes they want to make within E. coli or S. cerevisiae. And you can make thousands of different edits within an organism, but one edit per cell, but you can make a population of cells of thousands of different strains. After you've designed your library, or after you've designed the edits you want to introduce, the software itself creates optimum designs for introducing those edits. That initiates the manufacturing of the reagents and the consumables that are necessary to build that library. Once those reagents and consumables come in, you put them in another aspect of the platform, which is the Onyx instrument itself, which is shown right here. You put those consumables into the instrument along with the cells you want to introduce the library into, and you press go. And in a completely automated, hands-off way, the instrument takes care of building that libraries, which takes anywhere from 2 to 4 days to build it, depending on if it's E. coli or S. cerevisiae. So after 2 to 4 days, you get a tube of edited cells that come out of the instrument. Within this tube, each cell will have at most one of the edits that was designed within your library, but again, up to thousands of different edits. And that pool of edited cells can then go into your phenotyping workflow. Inscripta also provides DNA sequencing kits that allows you to determine the quality of that library that you've built, and then also genotyping kits that after you've done your phenotyping, which could be a selection workflow or a screening type of workflow, allows you to do DNA sequencing on those that hit population or those individual hits. That NGS data then gets fed into another software package called Resolver, and this is what determines the edits that's contained within those hits or the distribution of edits within your hit population. And this basically allows you to link the genotype to the phenotype. Then you'll probably want to take a hit strain from that first round and go through this cycle all again, and you can do really rapid rounds of genome engineering using the Onyx platform. We like to call this the Onyx platform really a genome engineering biofoundry on your benchtop. It creates optimum designs using the designer software. It utilizes the proprietary MAD7 nuclease, CRISPR nuclease. Designs per library for E. coli anywhere from 100 to 10,000, or yeast anywhere from 100 to 6,000. There's a single edit per cell. You can have a diverse edit types anywhere from 1 to 63 base pairs in length. And you can precisely deliver edits either genome-wide all over the place or at specific target sites within the genome. You can track the edit in every cell with barcode NGS. So there is a DNA barcode that's associated with every edit that allows you to do rapid genotyping by sequencing that barcode. It has push-button simple workflows, and it really democratizes genome editing. And I like to use myself as an example. So prior to coming to Inscripta, I had never designed a library, certainly never built a library, but I had phenotyped a library. But using the Onyx platform, I'm able to come up with an idea. I can very rapidly use the designer software to design that library. Then I can build that library using the Onyx instrument. And once I get that pool of edited cells, I can immediately go into my phenotyping workflow. So there's a wide diversity of edit types that can be introduced in both the coding and the non-coding regions. So in the non-coding region, you can imagine targeting regulatory regions within a protein. So this includes maybe the promoter or the terminator regions. You can target the coding region of the genome. We always like to do genome-wide knockout libraries. We do this by introducing a triple stop codon at the 5' end of a gene or all genes. You can also do saturation mutagenesis, where you mutate one residue of the protein to all other possible residues. So here's some more of the specs about the platform. So as I mentioned, the hosts that are currently supported are E. coli and Cerevisiae. This includes Inscripta standard strains and related customer strains. You can target the coding and the non-coding regions in the native genome, or if you've introduced heterologous sequences, you can also target those for editing. Designs per library, E. coli up to 10,000, Cerevisiae up to 6,000. You can have one edit per cell, but you can do insertions, you can do deletions, and you can do substitutions, anywhere between 1 and 63 base pairs. The library composition that typically comes out of the instrument after your library build, typically greater than 90% of designs are contained within that library of the original designs for your designed library. And the edit fraction is anywhere from 15 to 75%, depending upon the difficulty of the edits that you're trying to introduce. As far as time, reagent delivery is approximately 3 weeks, and library build is anywhere from 2 to 4 days, but your hands-on time with the instrument for a library build is really less than an hour. So the Onyx platform really accelerates scalable genome engineering and strain development. So how does this whole process work? So it all starts with building a base strain. This is where you've introduced your heterologous gene or heterologous pathway into your production strain. You develop a phenotyping workflow in order to assess the production of a compound or protein. Then you build a library on top of that, or multiple libraries on top of that, and we'll get some examples of how you do that and what type of libraries to do or use. And this is really where the strain optimization takes place. So use the Onyx platform to build a library, you phenotype that library, you select a strain to go into the next round, You take your hits from your first round and start combining them with one of the hit strains to combine diversity. And then you can go through this cycle multiple rounds in a very rapid fashion. And the Onyx platform really decreases the cycle time by up to 50% because the bottleneck at the design and build stage is dramatically reduced. And then after you've gone through enough rounds to develop a production strain with sufficient titers or quality, then that selected strain can go into for production. So the Onyx platform gives you increased access to the strain genome for making precise changes, and this accelerates the product to market timeline because you can go through these iterative cycles so rapidly. And increased sampling of novel variants can lead ultimately to better product performance. Okay, so I'm going to give you some examples of where we use the Onyx platform to improve a strain that can be used for production of two different proteins. And really, heterologous protein production is central to many different industries. And this, at its core, is this based on this concept that you can use microbes as sort of mini cell factories in order to produce massive quantities of proteins to be used in, say, industrial biotechnology space, biopharma or therapeutics. A really hot area right now is this alternative protein market, or applications in the agriculture space as well. So the first application I'm going to tell you about is where we wanted to try to improve the production of an enzyme called CBH1, which is important for the industrial biotechnology space, and we decided to use yeast as the host for this. So CBH1 stands for cellobiohydrolase 1. It's an important enzyme in the degradation of cellulose, and in fact, it's the most important enzyme in that degradation process. CBH1 is important for a process called CBP, or consolidated bioprocessing. And the CBP process has this idea that you have a strain that produces a suite of enzymes that are necessary for the degradation of some type of feedstock. So you mix this strain with this feedstock. The feedstock is often waste plant material, lignocellulosic plant material. So the enzymes that are produced by this strain, degrade that feedstock, producing sugars. The cells then, or that strain, will take up those sugars and then are able to convert that into some sort of fuel or some sort of chemical that can be used for an industrial application. So we decided to focus on a fungal CBH1 that was codon optimized for expression in yeast. We used the native signal sequence, the native fungal signal sequence, and we drove expression of the gene using the native yeast ENO2 promoter. So the base strain was built in SenPK, and we generated a single copy genomic integration of this expression cassette. So the assay is pretty simple and straightforward. It was using a PNP-based substrate, PNP beta-lactoperoxide substrate. CBH1 hydrolyzes this bond right here, releasing paranitrophenol, which has a characteristic absorbance at 405 nanometers. And by using an enzymatic activity assay, we're able to quantify the amount of active CBH1 that is being produced. Our screening workflow was all done in a high-throughput manner using automation. The microfermentations were done in 96-volt plates using 300 microliter of media. And to show you a little bit about the noise in this screening workflow, if I pick 96 colonies of the base strain and take it through the entire workflow, we have a CV of about 1.7%, so very low noise, which allowed us to identify CBH1 overproducing strains with even small improvements in CBH1 production. Okay, so now we had our base strain and our screening workflow. So now comes the fun part where we build libraries on top of this base strain and try to improve the production of CBH1. And we had lots of different concepts that we came up with of trying to improve production. But it really broke down into two different overarching themes, and those are using an unbiased approach where they're more genome-wide libraries, so targeting all over the genome, and then targeted or rational libraries. So for the genome-wide libraries, we generated a genome-wide knockout library, which had about 5,100 edits contained in that library, a genome-wide synthetic terminator insertion library. This is where we took a synthetic terminator and inserted it at the 3' end of every single gene in yeast, and ultimately this can have an impact on the overall expression of that particular gene. We also generated a genome-wide deletions library where there were deletions anywhere from 1 to 59 base pairs, and these were spread throughout the genome, targeting either regulatory regions or within the open reading frames themselves. So that's the unbiased libraries. As far as the targeted libraries or rational libraries, we had 2 libraries targeting the ENO2 promoter that was driving expression of CBH1. This includes insertions or deletions of the native transcription factor binding sites located within the ENO2 promoter, or swapping in other transcription factor binding sites at different locations within the promoter. I generated a CBH1 alternate codon library, and this is really a site saturation synonymous mutation library, where you take every codon and change it to all other codons that encode the same amino acid. So basically what we were trying to do was codon optimization in the context of the gene integrated into the genome. And then I have another library here that's called the targeted knockout library. And what I did for this library is I mined the literature looking for genes that have been previously shown that when you knock them out, they improve the production of a heterologous protein. I also did a genome-wide protease deletion library, so host proteases can often be problematic for heterologous proteins. And then I targeted some genes in the glycosylation pathway, which might have an impact on secretion of the proteins. So these are all the different concepts that we wanted to test. In total, there were 7 different libraries, which broke down into this unbiased and rational approach. So this is a summary of all the first round screening work that was done. As I mentioned, there were a total of 7 libraries. Between those 7 libraries, there were 15,000 different edits targeted, and I screened 8,500 isolates from these different libraries. They were all screened in a primary workflow where each isolate is screened as a singlet. Hits that we identified then go through a retest workflow where those hits are retested in quadruplicate to confirm that they're indeed hits and get some better measurement of the improvement that we're seeing. In total, there were 136 confirmed hits that we identified from these 7 libraries, and 74 of those hits were unique. So what I'm showing here is what happened, or the data from the first round, the primary screening data from the first round, the second round, and the third round. So that first round really provided the diversity that was used in subsequent rounds of iterative genome engineering. Excuse me. And I'm showing here the fold improvement over the base strain. So right here is the one-fold improvement. So this is the base strain activity. Here are the 7 different libraries that we identified in the first round, or that we screened or built and screened in the first round. And it was very satisfying for us to see that we saw nice hits from all of these different library concepts. So all the concepts that we came up with, we were able to identify hits from them, which is really satisfying and nice for us. So I took 2 hits from the first round, and those went into the second round. I stacked either a genome-wide knockout on those 2 hits, or I took all the hits from the first round, designed a new library, and stacked that on top of one of the hits. And that's what went into the second round, which is showed here. Very nice for us to see that in our second round screening, again, we're able to identify hits with significantly improved CBH1 production. I took the top hits from the second round right here. That went into the third round of screening, where again, I took that first round hit library and stacked on top of that hit from the second round, which gave us the primary screening data for the third round. Again, it was very satisfying to see that we continuously see improvement in CBH1 production through these 3 rounds of iterative genome engineering. Okay, so that's the primary screening data. Now what I'm showing you is the retest data for all the hits that we identified from the first round, the second round, and the third round. And I'm showing you the fold change in improvement in production over the base strain. And I arbitrarily picked about a 10% improvement as a minimal hit, or a minimal improvement in CBH1, to define it as a quote-unquote hit in our screening workflow. And you can see again here that from the first round to the second round to the third round, we're continuously improving CBH1 expression. These error bars here show the standard deviation of quadruplicate measurements, showing that there's very low noise in the assay, and that these strains are indeed improved over the previous round hits. Okay, so we had done a lot of unbiased approaches. So genome-wide knockout, terminator library, deletions across the genome from the first round, and then a genome-wide knockout stacked on first round hits. And I was interested in looking at what are the genes that we identified from this unbiased approach, and is there anything interesting about them? So in total, there were 69 different unique hits from the first and the second round that were identified in these unbiased approaches. And when I started to categorize them by different functions or different cellular processes, I found something interesting. So there are lots of genes targeting the protein degradation pathways, protein trafficking, several stress response. The most were actually in transcription translation processes, nuclear transport, a couple in the glycosylation pathway. 10 of them didn't really fit into any particular pathway, and they were kind of spread through various different cellular processes. It was also interesting to note that we identified hits in 11 genes of unknown function. So if we had taken a purely rational approach towards building these libraries, we probably would have never, or we personally would have never targeted genes of unknown function. But because we also took this parallel unbiased approach, this allowed us to pull at hits that we would have never identified from a purely rational point of view. It was also interesting to note that if you were to think about trying to improve protein production and where bottlenecks might arise, these bottlenecks would be in all of these different processes: protein degradation, protein trafficking, stress response, transcription and translation, glycosylation pathway. All of these processes are things that touch heterologous protein production. Okay, so the next thing that we wanted to do was to scale up these hits that we identified from our different rounds into shake flasks. And in order to do that, we took 3 hits from each one of the 3 rounds, and we scaled them up into 25 ml shake flasks. So we did our screening work in 300 microliters, but we wanted to see, can we scale them up to at least shake flask growth? So the first thing that we wanted to do was to measure the growth curves for these different strains relative to the base strain, which is shown here in black. And we were able to utilize a technology called a cell growth quantifier that was developed by SBI. And what this technology allows you to do is non-invasive, continuous measurement of growth in a shake flask or in other vessels as well. And it does this by measuring the backscattering from your cell culture. Continuously over time, we went up to whatever is 65 hours in this experiment. So it gives you nice detailed growth curves. And you can see here in black, this is the base strain, which interestingly had the highest amount of growth. And all of our different strains that we identified from the 3 different rounds, which I'm not going to go into the details of which ones are which, the important point is that all of them grew up to a lower degree. Some of them had shifted kinetics in far as how they grew, and then ultimately maybe plateaued at a similar level. Some of them had very similar growth kinetics, but then plateaued at a much lower level. It was interesting that all of them had decreased growth relative to the base strain. And this is kind of consistent with reports in the literature that they find strains that have improved heterologous protein production often have lower growth. And it's speculated this is likely due to the fact that carbon is, instead of going towards cell growth, more of that carbon is being diverted towards the production of proteins, which can be carbon intensive. Okay, so this is what the growth curves look like for all the different variants. But now we wanted to see, what does the CBH1 production look like? The more important thing. Again, these 3 hits from each round were grown in 25 ml shake flask cultures using the same media conditions that were used for screening. And this is what the data looks like. So we're able to look at the absorbance at 405 nanometers. Again, this is a measure of the amount of active CBH1 that's being produced over time. I'm showing the base strain here in this black dashed line at the bottom. It was very nice for us to see that the 3 first round strains that we picked all produced at CBH1 levels that were better than the base strain. Our second round hits are right here. Again, improvement over the first round hits. And then our third round hits were further improved over the second round. So it's very nice to see that all of the hits that we identified in our small scale screening that we scaled up, they all translated to larger scale. And in fact, these were all the hits that we tested at larger scale, and they all ended up translating to at least 25 mil shake flask culture. And the top hit that we identified here had about a 4-fold improvement in CBH1 production over the base strain. So a significant improvement in production. Okay, so that's a little bit about the type of improvement you can get. So we saw in this particular project, we saw a 4-fold improvement after going through 3 rounds of iterative genome engineering. Now the question is, well, how long does it take to do that? How quickly can you do it? And what type of resources, as far as FTE resources, are involved in doing that? So here's again a schematic of the overall workflow. So you build your base strain, you design your library, you build your libraries, you phenotype and genotype, and then go through this cycle multiple times. So the first round screening, this included 7 libraries, as I mentioned. We identified 74 unique hits from that. This whole process took us about 3 months. And I will say most of that time was spent doing the phenotyping, because the design and the building aspect has been completely debottlenecked to where that is really a small portion of the time. For the second round, which is a genome-wide knockout stacked on a first round hit, that was about 1.5 months. And the third round, which was a first round hit on top of the second round hit, that also took about 1.5 months. Again, most of that time being spent on the phenotyping workflow. So in total, in about 6 months time span, we're able to develop a strain with a 4-fold improvement in protein production by going through these 3 rounds of iterative genome engineering, the final strains having 3 edits contained within that production strain. And of course, you could keep going through this cycle multiple times, or many more times, to further improve the CBH1 production. As far as the amount of time, or resources, as far as people resources involved. So designing a library takes anywhere from, a single person, anywhere from a half an hour to a few hours per library, depending on the complexity. If it's a simple genome-wide knockout, you can use designer software, very rapidly allows you to do that. But if you want to design more complicated things like targeting the 2 promoter, that will take a little bit more time, as far as or outside of the designer software, in order to design those libraries and then upload those libraries into the designer package. As far as build, the hands-on time is really less than 20 minutes to start a run, and the run time is, in this particular case, is 4 days per library for SynPK. And the phenotyping and genotyping, which is really the most labor-intensive part, was about 0.25 FTEs in order to go through that part of the workflow. So overall, you're able to develop a strain with significantly improved production and phenotype in a short period of time, using really minimal amount of resources in order to do that. You really don't need an army of scientists to do this type of project. Okay, now I'm going to switch gears and talk about another project that we worked on where we wanted to improve the production of a gene called, or a protein called Asparaginase B. And this is really important for the biopharma and therapeutic space. And we decided to do this in E. coli using a B strain. So the enzyme asparaginase is involved in the conversion of asparagine to aspartic acid and ammonia. And this enzyme is actually important for cancer therapeutics. So it had been identified that several cancers are unable to produce asparagine, and they really rely on the asparagine that's circulating within the blood. So if you were able to find a way to degrade that asparagine in the blood or convert it to some other compound in the blood, you could then starve the cancer cells for this important amino acid that it needs for growth. And that's what was developed. This asparaginase is actually a candidate with broad use in various cancers, including acute lymphocytic leukemia, which has about 6,000 new cases per year. However, there are some improvements that need to be made with this asparaginase B, And a lot of them focus on production. So there's a need for improved production and purification of this important enzyme, and then also to overcome production toxicity. So this asparaginase it is somewhat toxic to E. coli. Overproducing it in particular is toxic. Okay, so the same type of workflow that we use for CBH1. The first thing that we did was we inserted the asparaginase gene into the E. coli genome. Then we designed different libraries against that base strain to produce edited cell library. We tested them in isolate, measuring asparaginase activity, so that goes into phenotyping workflow. Then you can do rapid DNA sequencing by the barcode, identify those hits, and then you could go through multiple rounds. So I'm only going to show the data from one round of this asparaginase genome engineering that we did. And again, we fell back to these two strategies to generate diversity. And you're going to hear this over and over again, and we use it for many projects, where we wanted to do an unbiased approach, which is genome-wide libraries. And then we also wanted to do a rational approach, which is targeted libraries, targeting genes that we think might be important for heterologous protein production in E. coli. So we did a genome-wide knockout library, knocking out every gene in E. coli. This was about 4,000 edits contained within that library. And then we did a genome-wide promoter insertion library. And in this case, excuse me, we took a strong synthetic promoter. So promoter is expected to have a strong increase in transcription, and we inserted that in front of every gene in E. coli. So those are our unbiased approaches. Again, just genome-wide. Then we had a targeted library. And the one targeted library that I'm going to talk about is what we're calling a cell morphology and protein folding library. And what was targeted was various different chaperones that can have an impact on the folding of proteins, proteases that obviously can have an impact on the expression of the protein because it can degrade, your heterologous protein can be degraded by host proteases, and then genes that are involved in morphology of the cell. And these different gene classes were targeted with a couple different edits, so a knockout library or knockouts of those genes, various different promoters that were targeting those genes as well as truncations of those genes. So those are the two library approaches, again, this unbiased and then a rational approach. So with E. coli, and depending on what strategy you're using, you can actually go through this design, generate, test, learn cycle quite rapidly. And it can be anywhere from 2 to 4 weeks, depending on if you're using a library that might be off the shelf. So like genome-wide knockout libraries, we routinely have those available. So you can very rapidly take your hit strain and stack a genome-wide knockout library on them. But anywhere from 2 to 4 weeks to go through this design, generate, test, learn cycle for this particular project. Somewhat faster than yeast. So I'm showing the primary screening data from these 3 different libraries, showing the fold improvement over the median, which is here at 1. We did pretty shallow sampling of only 441 isolates from these 3 different library concepts, but it was very nice that they were able to see some hits that had an even greater than 2-fold improvement in asparaginase activity. So again, looking back at the CBH1 story, looking at what type of cellular processes are these genes that we identified as hits, what cellular processes are they involved in. We saw 5 cell membrane genes. We saw 6 stress response genes. So this, again, is not necessarily surprising because heterologous protein production can be somewhat stressful to an organism, as well as we know that asparaginase itself is somewhat toxic to E. coli. The cells grow much slower when they're overexpressing asparaginase. We also identified 6 genes of unknown function. So we see this repeated theme over and over again. Again, because we took these 2 strategies of rational as well as unbiased approaches, it allowed us to pull out these genes of unknown function that we would have never identified with a purely rational strategy. And then we also identified 5 genes that are proteases or chaperones, again, completely making sense for heterologous protein production. Okay, so as far as conclusions, so the Onyx platform really enables rapid strain development for improved production for CDMO and CROs or for any other company that wants to develop production strains. It can increase the yield ultimately of that product through various different library concepts and various different cellular processes that you might touch on, that the libraries might touch on, and then also from a manufacturability standpoint, where you can decrease, say, product toxicity or overcome product toxicity like we did for the asparaginase project. And you can imagine designing libraries for E. coli where you're trying to decrease the amount of endotoxins that are being produced or ultimately end up in the final product, or decrease the, or increase, say, the tolerance to phage or any other strain property that is important towards scale up to production scale. The platform also allows for rapid workflows for significant reduction in both time and resources necessary to optimize production strains. So I talked about, for S-cerevisiae, for CBH1, it was about 6 months to go through 3 rounds with a 4-fold improvement. And for asparaginase, it was about, say, 4 weeks or so to go through this first round. And you can do that in just a few weeks to go through these cycles. And it also requires minimal amount of FTE resources because we've really debottlenecked the amount of time spent and resources or people resources that are spent doing the design and build aspects of these design, build, test, learn cycles. And the ability to sample greater amounts of diversity genome-wide really provides more shots on goal in identifying strains with better performance. Because you can make these very large libraries very rapidly, you have more opportunities of identifying hit strains that can have that improved performance. And also, one of the issues or one of the things you care about when you're going from high throughput screening, small scale, up to, say, lab scale fermentations, where the conditions are very different in those two, at those two different scales. The concern is always, do your hits scale up? Because the conditions are so different, and because you're able to identify many hits in this high throughput screening workflow by building much larger libraries, it gives you more shots on goal on identifying strains and hits from your small scale that will translate into larger scale fermentations. And with that, I'd like to thank you for your time and attention, and I'd be happy to answer any questions that you might have.
Thank you, Eric, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question here asks, can you talk about generate step more in detail?
Yes, so I think that's in reference to the actual building of the library, which happens on the instrument. So I can give kind of a brief overview of what happens on the instrument during the build process, but I would also encourage you to look at some really cool videos we have on our Inscripta website. There's a nice video called Onyx Instrument Tour that goes through all of the steps in a little bit more detail. But basically, for the generate, or what the generate part, or what happens on the instrument, is you put the consumables and reagents onto the deck along with the cells that you want to introduce your library in. You press go, the door will close. The instrument takes care of every step within the process. So it first grows up your cells to a proper OD that would be good for making them competent to do a transformation. So it grows them up. It makes them competent for doing the transformation. Then it mixes it with the DNA that's necessary to build the library, does the electroporation, recovery, and then it goes on to a specialized microfluidic module where the editing process actually takes place. Then it harvests those cells off of that module, collects it into your cell output tube, and then at the end of the run, you take that cell output tube, and then that's what goes into your phenotyping workflow. But that's kind of the basics of the steps involved in the build that happens all in the instrument.
Great, thank you. Next question here. Is it challenging to develop this platform for other organisms, such as Pichia? Excuse me.
Yep. Yep, we get that question a lot. As I mentioned, the platform currently supports E. coli and Cerevisiae as the two hosts. We definitely hear lots of interest in other organisms. So Pichia is one of the ones that we hear, and Bacillus is another one that we hear potential customers interested in. As far as it being challenging, I don't know if I would say it's challenging, but it definitely requires some development work in order to optimize the platform. There's a lot of work that has gone into getting optimized design scores for E. coli and for S. cerevisiae, as well as the DNA architecture that's necessary for getting that process to take place efficiently within the microorganism. So I don't know if I would say it's challenging, but it does take some development work in order to, or it would take development work if we wanted to expand it to other organisms.
Great. Next question here. What was your overall expression titer of ANSB in grams per liter?
Yeah, that's a good question. So we actually haven't measured the absolute titer for the asparaginase. The work that we've done has all been basically in small scale, high throughput screening. So I don't know what that would translate to if you went to larger scale fermentation, but we haven't measured the absolute titer yet for these strains. But good question.
Great, thank you. Next question here. You said that the CBH1 gene was optimized for expression in yeast. Why do you think you were still able to identify hits in the alternate codon library?
Yes, so that was actually an interesting library. So it was based on a idea that I had kind of I've wanted to test for quite some time, is to look at, when we synthesize codon optimized genes, is it really the most optimum gene structure for that heterologous protein? And I think the answer to that question of why we're still able to get hits out is that we don't fully understand all of the steps in translation, or how codon usage really translates into the translation of the ultimate protein. So I think one way to think about it is, there's been some reports in the literature that you can actually over codon optimize a gene, meaning that you can optimize it so that it expresses so rapidly that it doesn't have time to properly fold. And if you do, a complete codon optimization, that you can actually end up with less active protein at the end. So I think that's a potential part of it. There's also, you might imagine that you might want certain places within the gene where the ribosome might slow down or pause for a moment by maybe introducing a more rare codon at that location, and that will allow for the protein to fold, or what's been translated so far to fold properly. There can also be differences in tRNA pools at different times during fermentation, whether it be at the beginning or the end of fermentation, or in mid-log phase. So I think those are some reasons why, but I think it was very encouraging that we were able to get some nice hits out of that library, suggesting that there is still some improvements you can get as far as from codon optimization of the gene.
Great. Next question here. Do you think the genome hits identified for both projects will be generalizable to other proteins, or do you think that they are specific to those proteins?
Yeah, that's another question. That's good. So the answer is we don't know at this point. I think it would be really cool to do an experiment where we test multiple different genes, multiple different heterologous genes in these different strains to see if we see an improvement. I mean, I can imagine that some of the hits are probably going to be generalizable if there's things like, overall secretion of any protein, or transcription or translation or nuclear transport that might be important. But I could also imagine that there's certain hits that might be specific to certain proteins. So certain proteins might have specific bottlenecks, say for proper folding, that other heterologous proteins might not have. So I think the answer to that question is there's probably certain ones that are generalizable to all proteins, and then there's likely other ones that are specific to certain proteins, certain heterologously expressed proteins.
Great, thank you. Next question. Do you think that after several rounds of using the first round hits library and builds, you will eventually stop getting improved strains?
Yes. So the answer to that question is yes. I think ultimately, once you've gone through several rounds of this iterative genome engineering where you're using the same, the hits that you identify from that first round, those different hits might be touching on different bottlenecks within multiple pathways that are involved in heterologous protein production. And once you've kind of combined all those hits into a single strain, you probably will kind of exhaust all of the diversity or the improvements in diversity that you can get. And that's not necessarily the end of the project, because now what that means is that there's probably bottlenecks elsewhere that you might need to uncover and deal with. And what you often do in these types of projects where you're doing, whether it's protein engineering or genome engineering, you'll go through a series of rounds of combining your diversity into a single strain. And then you want to start adding new diversity. And what you do is you go back to one of your, say, unbiased libraries, so like a genome-wide library, genome-wide knockout library, and start adding more diversity again to your library. And that would allow you to uncover some new diversity that might have improved phenotypes.
Great. Looks like we have time for one more question here, so we'll wrap up after this one. And this asks, instead of using a single hit strain for the next round of iterative genome engineering, could you use a pool of strains for building the next round?
Yes. So the examples that I gave here, we used a single strain that went into the next round. It is definitely possible to use a pool of strains. So maybe you take 5 hits from your first round and stack, say, all the hits on top of that, or genome-wide knockout on top of that. That's entirely doable. And in fact, we have an app note or a tech note about that where we tested that very thing, and it definitely does work. There are some complications as far as using the software, the Inscripta Designer and Resolver software, in order to characterize those hits or those libraries that come out of it. So it's not, quote unquote, a fully supported workflow as far as from the end-to-end solution that we're providing with the platform, but it's definitely something that is doable on the instrument, and we have done that. And so if you're interested in more about that, I would encourage you to take a look at that tech note.
Great. Well, thank you again, Eric, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.